Okay, so as we all know, drug discovery process is a very long and very complex process in which a lot of economical burden comes into place. So computer-aided drug designing makes the process much easier as well as faster. So what is performed in computer-aided drug designing is that we use different computational tools to discover and analyze different kind of ligand molecules, which can be probably developed into a drug-like compound and further tested into different cell lines and move on to the clinical trial as well. So it is a basic process in drug discovery process, and which is very fast as well as reduces a lot of cost as well as time. So what CADD actually does in ADMET process as well, because in drug discovery process, a lot of drug molecules got rejected in different phases of clinical trials, or whatever you say like that. So in between that process, CADD minimizes the chances of failure in different drug development procedure. So in CADD computer-aided drug designing, there are two basic approaches. One is structure-based drug designing, and another is ligand-based drug designing. In structure-based drug designing, the particular target or receptor, which can be for example, protein or enzyme, the structure of that target protein or enzyme is known. Whereas in ligand-based drug designing, we don't know the structure of target protein. This is a overall procedure of CADD, in which first you need to find a particular target. So after identification of target means a receptor protein. So after identifying this target protein, if the 3D structure is available, then you can directly move on to structure optimization. Whereas if the 3D structure is not available in database, then you need to go through homology modeling. So homology, for this particular homology modeling session, later on, Mr. Rabin will tell you much about homology modeling. And after 3D structure, if a 3D structure is available in the database, then you can directly move on to structure optimization. And further, you need to identify the particular binding site of that protein molecule or enzyme, and then you move on to molecular docking procedure. In molecular docking procedure, what exactly is done is, I'll later on show in upcoming slides. Basically in molecular docking, the ligand molecule is docked into the particular active site of receptor molecules. After that, different sorts of optimization and structure minimization would be done using different sorts of algorithms and scoring function. And after that, the particular conformation, the conformation which is fit into the ligand, which is fit into the receptor, which is fit into the active site residues, is selected, and further it moves on to producing heat molecule. So if the molecules are probable to be a heat, that means if it binds to the particular active site, producing different hydrogen bonded bonds or different other biological bonds, then it is taken to further molecular simulation, dynamic simulation, and then various wet lab analysis would be done. For example, biochemical assays, different biochemical assays. If a particular drug function in particular biological property, then it is tested into that particular different biological assays would be performed at that step, and then further QSR and ADMET analysis can be done later on after that confirmation, and finally that particular lead molecule would be optimized, and then clinical trials would be performed. So it shows the structure-based drug designing, where a ligand molecule, it is a ligand molecule. You can obtain this ligand molecule through organic synthesis, or for example, in various natural product, you can isolate the compounds, and different approaches are there, such as NMR and different mass chromatography are there. So you can do this chemical procedure to synthesize different molecules. So after that, or simply you can prepare this ligand molecules through biochemistry screening, and then the prepared ligand molecule are further docked into the particular receptor molecule. So receptor can be obtained through database. Structural biology means it is either obtained from various databases, or it can be if the structure is not known, then the structure can also be prepared through homology modeling. And finally, the docked structure is obtained using different molecular docking approaches. And then finally, we can create a potential drug molecule, which is taken into clinical phases, clinical trials. So here it comes to molecular docking. You can see a particular ligand molecule docked into the particular active site of this receptor. It is molecular docking is simply a computational tool in silico method, which is used to find the interaction of ligand molecule into the particular active site of DNA or either protein or even enzyme molecule to form a biologically active complex molecule. The biologically active complex molecule is further taken for different biological activity. And this molecular docking is generally is done for conformation of a particular conformer, which is exactly fit into the particular active site of receptor molecule. And it uses different sorts algorithm, Lamarckism genetic algorithm or various genetic algorithm, and different force fields are inserted there, which are known as scoring functions. And this is a simple molecular docking procedure. It's a receptor protein. You can see a particular, you can see the active site of this receptor molecule. It is a single active site. You can have a lot of allosteric site there. So while performing molecular docking, you can perform in different sites. What you need to do is you need to find the minimum binding energy of that ligand molecule in the receptor protein. And this is a ligand molecule. When these two are present, receptor protein and ligand molecule, and then you can dock this ligand molecule in the active site of receptor protein to find this conformation. And there are different techniques involved in molecular docking. For example, ligand or receptor molecule is kept fixed. They cannot be moved. So it is a rigid ligand and rigid receptor docking, whereas there is flexible ligand and rigid receptor docking. Similarly, flexible ligand and flexible receptor, or rigid ligand or flexible receptor docking. So you can see this figure where a ligand molecule is present in the active site of this receptor protein, and you can see a particular hydrogen bond. So once you get a hydrogen bond present in this ligand molecule with the interaction of amino acids of this receptor protein, you need to know that this is the physical ligand molecule, and it can be further analyzed. There are different programs which are used for docking processes. Generally used is autodock. In autodock, generally now it is used autodock BINA, which is genetic algorithm, Lamarckism genetic algorithm, and it is very simple as well. Apart from autodock or autodock VINA, there are a lot of other tools. Actually, autodock VINA is freely available, so mostly people use this. Mostly students and other researchers, they use this autodock VINA. Whereas there are a lot of other programs where anybody can select this kind of programs to perform molecular docking. And let's move on to ADMET. ADMET is a in silico pharmacokinetics, where mostly according to FDA, it is said that oral route is the most used route for drug administration. So in oral route, we test for different properties of the drug, such as this is absorption, this is distribution, metabolism, excretion, and toxicity of the drug. So various parameters are analyzed in ADMET analysis, and this process is the process where a ligand molecule can be recognized as a drug-like molecule. So what we exactly do in this process is that simply anybody can do this procedure by comparing the criteria of Lipinski rule of 5, where any kind of ligand molecules should follow this 5 categories, should follow this 5 criterion. So this molecule or this ligand molecules can be considered as a drug-like molecule. For example, the ligand molecule should have less than 5 hydrogen bond donor or less than 10 hydrogen bond acceptor. and the molecular mass or molecular weight should be less than 500 Dalton, and the lipophilicity constant, lipophilicity value should not exceed 5, or even molar refractivity value should be between 40 to 130 angstrom square. So these all are the things we use to check the drug likeness of any particular ligand molecule. So this figure shows the absorption. This is a particular structure that is a erythrocyte present here. So when drug molecules goes into the body, the absorption takes place here. So for absorption, topological surface area is used, where the topological surface area should be less than 140 40 angstrom square, which means the drug molecule should be lower in polarity, should have lower polarity, so that it can be easily absorbed into the body, different cells or tissues of the body. And in case of distribution, the drug molecules does distribute in the different part of body. So for distribution, value of distribution VDSS is analyzed here. So there are different parameters and their values. So any ligand molecules that is considered to be a drug molecule should follow this kind of different parameters, the values of different parameters. So that in distribution, that central nervous system permeability, CNS permeability, or even blood-brain barrier are analyzed over there. And when we move on to metabolism, so usually in metabolism, cytochrome is generally used as a indicator. So the level of cytochrome present in here can be analyzed for this metabolism of body. As well as in excretion, we analyze the total clearance done by kidney or liver or even lungs. So there are different organs that plays role in excretion. So the total clearance value is observed there. And in case of toxicity, MS test, in silico MS test is done, which is generally used for this toxicity analysis. So there are various online tools for knowing the ADMET properties of any ligand molecules, such as PS, GastroPlus, Swiss ADME, FabDrug4, PKCSM, WolfSub. And generally, I've been using Swiss ADME and PKCSM. I usually use this because I found it much reliable than other sources. So I use Swiss ADME or PKCSM. Later on in hands-on training, we'll provide all of the information, and we will do all of these things in online as well. So after that, it is a pass. So what exactly pass is that if you have a ligand molecule, if you synthesize a ligand molecule, of if you obtain ligand molecules through partial screening, and you don't know exactly where it get on to bind and perform different biological function, then you can simply use this pass tool. Pass tool actually performs or actually give result based on the spectra, the structure of that ligand molecule. So this pass has a less than or equal to 95% of accuracy. So it predicts the particular receptor site, a receptor or protein where the ligand molecule can get bind into. So you can directly go to pass online and you can perform this. So I've put on this slide for your age, such as it is aspirin molecule. So I have provided this aspirin molecule prediction, and the result are given here. So how can we analyze this then? So in case of pass online, it gives a result here. So what is PA and PI? So PA is a probability of being active into that particular receptor, and PI is a probability of being inactive into the particular receptor. So while choosing any receptor molecule for a particular ligand, you need to have a value of PA lesser than greater than 0.5, or it is actually said that 0.6 or 0.7 would be a better one for ligand for molecular docking process. So you can see aspirin can directly bind to fibrinocyte receptor. So you can go to different database, and you can easily extract the PDF file and then move on to molecular docking, or either you can do homology modeling for that particular receptor. That's all. And you can see different databases and tool for receptor as well as ligand molecules. So in case of receptor enzyme, you can use protein data bank, which is normally worldwide used famous database for generating protein, different structure, crystallography structure of protein. And in case of ligand or drug, you can go to PubChem and extract any ligand or drug molecules. Drug being also there for this ligand or drug molecule. And another is for structure and format conversion of ligands. So in case of structure making or format conversion, you can use ChemInfo. I've given the link as well. You can use ChemInfo. You can go to ChemInfo and put on the name of that particular ligands. You can get the structure, or you can either make the structure and get the name of that particular ligands. And OpenBabel is another online tool which is used for format conversion. There are a lot of formats used in ligands, such as I'll tell you later on. But for example, SMILES STA format are there, so you can easily convert using OpenBabel. Or in the people who are studying organic chemistry, or who are from the field of organic chemistry, they know this ChemDraw. So you can either use ChemDraw as well for structure making, or simply drawing a structure. And so in a receptor molecule, there is an active site. So how can you predict the active site and the particular active site residues then? For that, we use CAST-P and PDB-SUM. So for CAST-P, the CAST-P is one online tool, freely available on the online tool. You can go to it. So there's link, and you can directly use this CAST-P. So I've given an example of one protein molecule here. So in case of CAST-P, what we do is we get the PDB ID of a particular receptor molecule, in which we need to find the active site. So you can directly paste the PDB ID of the protein interest. So you can directly go there and get the pocket or active site of the receptor. This receptor. So another tool is for so it is that. So by using CAST-P as well, you can find the active site residues. But another tool is there. It is PDB-SUM. So you can go to PDB-SUM and put the PDB ID of particular receptor molecules, and you can get if it has already a parent ligand, then you can get a particular active site residues with a parent ligand. For example, it is a acetylcholinesterase enzyme where galantamine is inserted there. So galantamine is a particular enzyme, particular ligand present in there. So galantamine is a acetylcholinesterase, anti-acetylcholinesterase ligand. So you can see the active site residues present over there. For example, glutamine, serine, many more. You can see the interaction here is a hydrogen bond. You can use this. So another thing is that file format. File format is normally for ligand molecules. So during molecular docking and different CADD procedure, you need to you use different kind of file format. For example, SMILE file format, SDF, MOL, PDB, PDB-QT. So SMILE, SDF, MOL, and PDB is for ligand molecules, whereas PDB-QT is a file format generated during molecular docking with Autodoc or Autodoc Bina or any other tools. And you can use molecular visualization tool, for example, Chimera, PyMol, and Discovery Story Visualizer. Most easiest one is Discovery Story Visualizer. You can either use PyMol or Chimera as well for molecular visualization. Even you can perform modeling as well, modeling in this different platform. So this is all about basic information on CADD. So if you have any question, you can ask me, or we can move on to next presenter. We'll be presenting homology modeling. After this process, you will be presenting homology modeling. If you have any question, you can ask me. Sorry, it's a recurrent meeting, so you can join. You can join through the same link, and we'll have another presentation. So once it's gone, you can join this meeting after all. Okay, if you have any question, you can ask me. The floor is open. Okay. I didn't find any queries about my presentation. So let us welcome our next presenter, Mr. Rabin Pahari. So he has been working in dengue. So he will be presenting on homology modeling. Rabin, now let's stop
Thank you,
sharing.
Suman.
So we have less than 8 minutes. So after the end of this, you can simply join on through the same link, and we will be moving forward. Okay, Rabin, the floor is yours.
I'm unable to host disabled. Can you enable for screen sharing for host?
Okay.
Hello, everyone. Thank you, Suman, for having me. Today we are going for the webinar in computer-aided drug design, homology modeling part. So what is the process of computer-aided drug designing? Suman has already told about the processes, these processes. So I'll be only talking about the homology modeling part. As you can see, I have a circle down this homology modeling part. So why homology modeling? Development of molecular biology, rapid identification, isolation, and sequence of genes. This gives a lot of data, and this consists of different databases like NCBI, UniProt. So that database consists of different structures, sequences. So why we should do homology modeling in the context of developing country like Nepal? We do not have the resources to conduct X-ray diffraction NMR for obtaining the 3D crystal structures. So in that case, we can use homology modeling, which is a software-based approach to obtain the 3D structure of proteins. Also, similarly, we do homology modeling for that proteins that do not have 3D structures already. So in case of that condition, we should do the homology modeling. Homology modeling is basically the method that can be applied to generate reasonable models of protein structure. What does homology modeling do? It predicts the 3-dimensional structure of a given protein sequence target that can be obtained from the different databases like UniProt, where we can simply obtain the sequences in FASTA format and simply input in the homology modeling server like Swiss model. It can target based on alignment to one of the more known protein structures templates. So in case of example, I'm taking the example of Swiss model. It has SMTL, Swiss model template library, where it consists of various 3D crystal structures of the proteins, similarly structured proteins. So we can align them with our target protein and create the models. If similarity between the target sequence and the template sequence is detected, structural similarity can be assumed. In generally, 30% sequence identity is required for generating useful models. So let's go on the main homology model building one of the freely available server which can be available at swissmodel.xphagy.org. It is relatively easy than other homology model softwares. In this homology modeling workflow, you simply have to pick up the target protein sequence from the databases like UniProt in FASTA format or their ID. After taking the target protein sequence, you need to input into their server, and it will automatically align with the template library, SMTL library. So it will give the target template alignment. After obtaining the target template alignment, after obtaining more than 30% sequence similarity, we can build the models. And further models should be evaluated through QE tools. And further model evaluation should be done by other energy analysis tools, and similarly molecular dynamics simulation. So we are not covering at the moment molecular dynamics simulation. We only how we can build a simple model. We are focusing on that at the moment. So after inputting the after sequence into the Swiss model server, you will get the model in PDF file format, which can be visualized by different softwares like PyMol, Chimera. So here is the model of dengue virus I've done in my thesis. So how does model will be evaluated? This is the main thing we should consider. The terms like GMQE and QMIN. Quality gives the quality estimation. GMQE means global model quality estimation, which will be ranked from 0 to 1. Higher is better. Similarly, QMIN, quality model energy analysis. It will also be in between 0 to 1. Higher will be better. This is the Swiss model obtained from that Swiss model server. Homology model of dengue virus to full length NS2, NS3 protease. So this is all. Over to Suman.
Okay, so we have just 1 minute left for remaining portion. So if anybody has any question about this particular session, you can put on your queries. Later on, we are having another Q&A session in another session. So if anybody has any question, please. Okay. Thank you for everyone for participating our webinar. So let's have some Q&A session after this. So you can directly go to the